Okay. Okay. Sure. Yeah. All right. Share. All right. So now it's going to be microbial nutrition and growth. And what we're talking about here is the way that bacteria grow when they have enough to eat and what happens when they run out of that, and also what they eat, right? What do bacteria live on? Different bacteria can survive on different types of nutrients. Okay. There are some bacteria that don't require hardly any nutrients. They just require the same kind of things you put in fertilizer in the soil, right? Some nitrogen, some phosphorus. Okay. And then they use photosynthesis to make what they need to generate ATP. All right. But a lot of the bacteria that cause human disease live off of components that you'd find in the body. Okay. So they're going to feast on um, material that's part of cells and material that's outside of cells. And so I'm going to talk about that as well. Okay, so let's do a new share. There we go. All right. So we're going to talk about what bacteria need, talk about how they get their building blocks and where they pull their energy from. We're going to talk about conditions under which a cell can survive. Okay. And here we're talking about osmotic conditions and different pH conditions. And then we'll talk about some cell transport as well. So when we're talking about any living thing, all right, we have what are called essential nutrients, which have to be provided to an organism for it to survive. And then there are macronutrients, which the organism needs in large quantity in order to live, and micronutrients, which are needed, but in smaller quantities to allow the organism to live. Okay. So micronutrients would be things like minerals. Okay. Macronutrients, um, for us, macronutrients would be things like lipids and proteins and carbohydrates, and water can be considered a macronutrient. And for a lot of bacteria, that's also true. Okay. So an inorganic nutrient is one that doesn't have carbon and hydrogen in it. Okay. And that would be something like sodium chloride or um, calcium phosphate could be some examples of inorganic nutrients. They're found in the Earth's crust, in water, and in the atmosphere. Okay. And for an organism to have those inside of it to function, the organism needs to consume it, needs to eat it. Okay. Same way we get our minerals and our vitamins, we eat them in our diet. Organic nutrients have carbon and hydrogen in them, and they're going to provide energy because of the carbon hydrogen bond. Okay. So the carbon hydrogen bond is high energy. Okay. a lot of organic nutrients are polymers, such as complex carbohydrates, proteins, and to a lesser extent, nucleic acids. Okay, and a polymer is simply a repeating chain of monomers. Okay, and in this case, the monomers are certain molecules. Okay, so the monomers of proteins are amino acids. Stop it. Hold on. No. There we go. So amino acids. Okay, the monomers of carbohydrates are simple sugars. Or you could say monosaccharides. Okay, the monomers of nucleic acids like DNA and RNA are nucleotides. Okay, so different building blocks. Okay, water is 70% of all living things. Organic compounds are 97% of the dry cell weight. And proteins, as we've talked about earlier, are the tools of life. Hold on just 1 second. All right. The elements carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur make up 96% of dry cell weight. Most chemical elements are available to the cell as compounds as opposed to pure elements. Compound means it's just chemically bonded to some other element. Okay, like the way that we get sodium and chloride is as sodium chloride, table salt, and not as just pure sodium or pure chlorine. Okay. Only a few types of nutrients are needed to make all the different compounds necessary for life. Okay, so you can see in the table there what's going on. Now, we can define an organism by what it eats. Okay, and why is this important? Okay, because this can allow us to identify how we can grow it. Okay, and also how we can kill it. So that's for growth and kill data. Okay, so that's why this is important. A heterotroph is something that has to obtain carbon in an organic form. Okay, so meaning what? As carbohydrates or as protein or as lipid. Okay, or in some cases in bacteria, you can even do things like methane and other hydrocarbons. But that carbon hydrogen bond is in all of these. And so that provides a lot of power when you snap it. An autotroph is something that uses carbon dioxide as its carbon source and can convert the carbon dioxide into the stuff that we listed for a heterotroph, okay, into organic compounds like you see up here, all right? So it doesn't depend on other living things to survive because it can either use light energy to do this in the process known as photosynthesis, or it can use certain compounds that are not organic to provide the energy, and we call that chemosynthesis, okay? A phototroph is any microbe that can photosynthesize, okay? And again, remember that photosynthesis is taking carbon dioxide and water, and then the energy from the sun, and making sugars and oxygen, okay? That's photosynthesis. The chemotroph gets energy from certain chemical compounds that would substitute for, in this equation, the energy from the sun, okay? So a chemotroph, for instance, might be able to pull energy from certain pure metals, all right, to do this. So it's interesting that bacteria can do tricks that we can't. A photoautotrope is photosynthetic, right? And we already talked about what that involves, okay? So they have to have light to exist, okay? Chemoautotrope uses organic compounds for energy and an inorganic compound as a carbon source. And a lithoautotrope relies on inorganic minerals and requires no sunlight or organic nutrients because it makes it itself, okay? So where's it getting its carbon? It's getting its carbon from CO2, okay, carbon dioxide. And that's in the atmosphere, okay? So these guys are totally independent nutritionally, totally independent from other life. Okay, that goes for the lithoautotropes and the ones that can do photosynthesis. Okay. A chemoheterotroph is like us. Okay, it gets its energy from an organic compound, a carbon hydrogen compound that already has energy in it. Okay, and what these organisms do is they do respiration or fermentation. Okay, so what's respiration? Well, respiration, the overall equation is going to be C6H12O6. That's carbohydrate plus oxygen gives us CO2 and water, okay? And if we were to balance this equation, there'll be a 6 there, 6 there, and let's see, 6, 8, 6, 12, so I believe it's 6 oxygens, right? Yeah, pretty sure that's right. It's 12 and 6 is 18, and that's 12. That's 6 and that's 18. Yeah, that's the balanced equation for respiration. Okay. Fermentation is a little different. Fermentation can sometimes produce ethanol, and sometimes it can produce lactic acid, and we'll get to that a little bit later. Saprobes are living organisms that feed on organic detritus from dead organisms. So basically they eat the dead, okay? Like rotting logs that can sprout mushrooms. Mushrooms are saprophobes, okay? So they decompose plant litter, animal matter, and dead microbes, okay? They're recyclers, all right? A parasite gets its nutrients from a host, okay? While a pathogen can cause damage or even death to the host. And could be anything from a virus to a helmet, right? So pathogens can be living or non-living. Okay, so we've seen non-living pathogens already as viruses and as prions, okay, and even as naked DNA, okay, in some cases, right? Examples of non-living pathogens, okay? Toxins are non-living pathogens. Ectoparasites live on the outside of the body. Example of an ectoparasite is a tick, okay? An endoparasite lives in organs and tissues. An example of that would be a tapeworm, okay? Or a liver fluke, right? Those are examples of endoparasites. Intracellular parasites live inside of cells. Examples include the leprosy bacillus and the syphilis spirochete, okay? So those are interesting, right? The leprosy bacillus damages nerve endings, so you can't feel pain. And then when you get a wound that becomes infected, you don't treat it because you don't feel it. And then that tissue becomes necrotic and eventually falls away, okay? Even on a living host. The spirochete for syphilis is a little spring-shaped bacteria, okay, that can move through cells. And when it does that, it leaves traces of itself behind on the cell surface that distract our immune system so that our immune system can't catch it, right? And the end process of syphilis eventually gets to brain damage, right? So before that, you have several other stages. You have a sore that eventually heals, and then you end up with symptoms of initial venereal disease, and like pain and damage to the urogenital system, and then eventually it can spread all the way to the nervous system. Okay? An obligate parasite cannot grow outside of a living host, while some parasites have a stage where they can live as free living organisms. Okay? So this is just a list of the essential nutrients, right? What's in most fertilizer, right, that you let plants grow in is nitrogen, phosphorus, and sulfur, okay? And the plant then uses photosynthesis to grow, right? Because these are all building blocks that it has to get from the water that's in the soil through its roots, okay? And then it produces for us oxygen and sugars, okay? And then the other organisms live off what the photosynthetic organisms make, right? For in most ecosystems, the photosynthetics those are what are called producers, right? And what feeds off the producers? Consumers, okay? And then the consumers and the producers all eventually die, okay? Eventually life and then death, right? Life and death. And then in death, we have decomposition. Okay. And that's things like saprophobes and bacteria and so on. And then all of those goodies, those organic and inorganic components, go back into the soil and into the atmosphere, and then the whole thing starts all over again. Okay. Sodium is important for cell transport. Calcium stabilizes the cell wall of endospores of bacteria. We need it for our bones and for blood clotting and muscle contraction and nerve transmission. The bacteria use it for other things. Magnesium is part of chlorophyll and also stabilizes ribosome function, and also stabilizes membranes. Iron is part of respiration. Okay, so iron is part of what makes ATP, and in many cases, oxygen. Okay, not oxygen, consumes oxygen, makes CO2. My fault there. CO2. Okay. Zinc is important for gene expression in things like protozoa, okay, because it's used by the enzymes that work on DNA in order to make and repair DNA, okay? So you can think of it like a separate tool that tool of life needs, like a plumber needs his wrench. Okay? Now, the way that we get stuff into the cell is through transport, right? And transport comes in two forms. Passive transport does not require energy, okay? So passive transport. And active transport that requires energy, okay? So no energy needed. Okay. And here needs energy. Okay. The driving force of transport is atomic and molecular movement, okay? And diffusion is an example, right? In diffusion, what happens is that material moves from high to low concentration without any input energy, okay? And that happens naturally. You can see that in, like when you put a drop of food coloring in a glass of water, okay? And it'll eventually spread throughout the entire volume of water. Or if you see a puff of smoke and you wait, and eventually it'll diffuse, right? It'll populate all the air in the area, okay? And eventually all the air in the world, and be evenly distributed, okay? And that happens without an energy input. And then there's the movement of water down its concentration gradient, which is called osmosis, okay? So let's listen to a little bit of osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, There is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So if you want to boil this down to a simple phrase, all right, we could say water chases solutes. A solute is whatever is dissolved in the water. And you know something's dissolved in water if it's clear, right? So wherever the solutes are, the water is going to run to it because the water concentration there is less. Okay. So we can see examples, right? In a hypotonic solution, like here, the rate that water moves in is greater than the rate that water moves out, and the cell will swell up. Okay. In a cell that doesn't have a cell wall, that's going to cause lysis. Okay. But in a cell with a cell wall, that'll cause an increase in volume, but the cell doesn't necessarily die. All right. So it'd be like a bacterial cell or a plant cell or a fungal cell. Okay. In a hypertonic solution, the cell will shrivel and may die, and that's called crenation. Okay. And the same thing happens to a bacterial cell. It will shrivel and may die. Okay. You're basically sucking the water out of it. You're dehydrating the cell, right? In an isotonic solution, function is preserved, meaning it continues to just do its thing, right? And that's because the rate at which the water moves in and out of the cell is the same. Okay. So what we always shoot for, right, if we're a living organism, is an isotonic situation. Okay. In our body, there are body systems that do that, like the urinary system, the digestive system, the respiratory system, and so on. Okay. In a single cell, the way that we deal with that is to run towards an isotonic situation through movement, right? Like chemotaxis, okay? Okay. So paddle away from the bad environment, paddle towards the good one, okay? Other transport processes, okay? We talked about diffusion, right? Simple diffusion is just movement from high concentration to low concentration with no energy input needed, okay? In facilitated diffusion, you move from high concentration to low concentration, but you need a helper to do this, and the helper is usually a membrane protein, okay? A membrane protein. And the reason for this is that for the material to move across the membrane, if it's charged or if it's polar, it has to have a special gate, tunnel, or pump, okay? Here, that would be a gate or a tunnel in facilitated diffusion. Okay. And then in active transport, energy drives movement against the gradient. Okay. And the energy can be in the form of ATP, or it could be another gradient. Okay. That's generated from using ATP, right? So something you want to move in a direction it doesn't normally move can hitch a ride on a different gradient that does want to move in that direction and get pulled in or out of the cell. Okay. An example of this is how a lot of cells get sugar inside, right? You use something called a sodium glucose symporter, where the sodium, which is high concentration outside the cell, and the glucose, which is low concentration, they both bind to the same membrane protein. And then because of the driving force of the sodium gradient, the sugar gets pulled in. Okay. So let's take a look at active transport. Where are you at? Come on. Oh, shoot. Yeah, I know where to go. Yeah. Hold on. Oh, there it is. It's there. All right, just have to go below it. I got you. I got you. Low. Well, shoot. All right, we'll do it. We'll do it this way. We'll do it the clunky way. Like this. Sure. Second desktop. Right. So here we go. There we go.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy for ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and to push the sodium ions across the membrane, where they are released outside the cell. The potassium ions now bind to the transport protein, and then the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay. Bump up. Remote share. There we go. Okay. So this is driven by energy. Energy drives the movement. Okay. Then we have what's called vesicle mediated transport. Okay, vesicle mediated transport. Okay, meaning what? Membrane bubbles. Stop it. Hold on. Got all my stuff. Oh. Membrane bubbles. Move material. Move stuff. Okay. And you need ATP to do this. You need energy. Okay. So this is active transport. Okay. So in endocytosis, a portion of the cell membrane just wraps around something and pulls it inside. Okay. In phagocytosis, the material is solid, and in penocytosis, the material is a liquid. Okay. So solid matter here, okay, liquids here. Okay? It's the same process, okay, in terms of what's going on. Right? It's just that the choice of material is different. Okay. Talk about stages of growth and factors that affect it. Okay. And talk about biofilms. Okay. So every living thing has what we call an optimum environment. Okay. Which means the best conditions to live. Okay. So what are some things that go into the optimum environment? The best pH, the best temperature, the best osmotic concentration, okay, the best pressure, okay, and the best amount of nutrients. Okay. And those conditions provide for the fastest growth equals fastest growth. Okay. So for bacteria, really for anything, we can have these temperatures, right? Cardinal temperatures are measurements for growth at a given given microbial species. All right. The minimum temperature is the lowest temperature that permits growth, but below this temperature, the bacteria can't do much. Okay, they may even die. The maximum temperature is the highest temperature at which growth can continue before the cell basically gets cooked, okay? So you got your low temp and your high temp, okay? And that's the window of life, right? Right, so you could set a window here. You got minimum, temp, you got maximum temp, and then somewhere in between, you're going to have what's called the cardinal temperature, which is the best temperature. Okay, and above and beyond this, you're going to have death. Below this, you're going to have death. Okay. So there's your optimum temperature, right? Right in the middle. Temperature between the minimum and the maximum that provides the fastest rate of growth and metabolism, okay? So we could say, we could rename this optimal temperature. We want to be as correct as possible. Let's do that. So optimum temperature. Okay. For most organisms that cause human disease, the optimum temperature is equal to body temperature, which is 37 centigrade. All right. That shouldn't be a surprise, right? Because that's where it makes its living, is inside the human body. And that's also true for a lot of microbes that live on us, in us, and around us that don't cause disease. They like body temperature, okay? Now, some organisms go outside those ranges, right? A psychrophile has an optimum temperature below 15 and can grow at 0 centigrade, which is temperature where ice freezes, or where water freezes into ice. So it's obligate with respect to cold and cannot grow above 20, right? Storage at refrigerator temperature incubates them and lets them grow and doesn't keep them from growing. Natural habitats of psychrophilic bacteria, fungi, and algae are lakes, rivers, snowfields, ice, and the deep ocean, right? Place where it's cold. And they usually don't cause disease. Well, that makes sense, doesn't it? Right? Why would something that likes to grow at well below body temperature cause human disease? It wouldn't, okay? Psychrotrophs grow slowly in the cold, but have an optimum temperature between 15 and 30 degrees centigrade. So these guys have some potential for disease, okay? Staph aureus and listeria are able to grow at refrigerator temperatures and can cause foodborne disease, okay? Mesophiles are the ones that cause most human disease, okay? And they grow between 20 and 40 centigrade, and they live inside or on animals and plants. You can also find them in the soil in temperate, subtropical, and tropical regions. Where don't you find them? In polar regions, right? Too cold. They're not going to get on there, okay? Human pathogens are optimal between 30 and 40. So 37 is body temperature, and that is where a lot of them really do their best. Thermoduric bacteria can survive short exposure to high temperatures, but normally grow between 30 and 40. And there are often contaminants of heated or pasteurized food, such as Clostridium botulinum and bacillus, right? So examples of diseases caused by this: botulism, Okay. This is why if you ever find a can in the grocery store that's doesn't look like a can, it looks inflated, like if the can has a puffy top or maybe even a puffy bottom. Okay, you don't buy that. That's contaminated. Okay, you throw it out. The reason is that the bacteria grown in there make gas, and the gas plumps out the can. Okay. A thermophile grows at temperatures above 45. You find them in the soil and water. And they're usually associated with volcanoes, compost, and habitats that are directly in the sun. You can also find them near hydrothermal vents in the floor of the ocean. Okay. So they dig really hot temperatures. They can grow between 45 and 80. Okay. Most eukaryotic pathogens and non-pathogens can't survive above 60. Okay. So these thermophiles are generally either bacteria or archaea. Okay. Extreme thermophiles grow between 80 and 121, and the same deal, right? They are going to be either bacteria or archaea. Okay. I mean, that's hot. That's very hot. Okay. If you go on Google and convert 80 centigrade to Fahrenheit, you'll see how hot that is. Okay. You're talking like being in an oven or a kiln. Okay. All right. How about the gases that we need, right? Oxygen and CO2 are important. Oxygen has the greatest impact on growth for microbes. It's an important gas, and it also is an oxidizing agent, meaning that it can strip energy and electrons from other molecules. Okay. Microbes can be either those that use oxygen and detoxify it, those that neither use oxygen or detoxify it, and those that don't use oxygen but can detoxify it. Okay. So oxygen enters cellular reactions and is transformed into singlet oxygen, which is a toxin, it's bad, and superoxide, which is also dangerous, and hydrogen peroxide, which is toxic. Okay. So all of these are bad news, as are hydroxyl radicals. Okay. A lot of cells have enzymes that scavenge and neutralize oxygen byproducts. Okay. So some examples of this are superoxide dismutase that can take oxygen and some protons and make hydrogen peroxide and water and oxygen. And then hydrogen peroxide can be broken down by catalase into water and oxygen. Okay. And these guys are non-toxic. Okay. And that's where we want to get to non-toxic. All right. So in bacteria that have this, when you put hydrogen peroxide on them, they will bubble. Okay. And they will be making oxygen bubbles. Okay. Which means that they have catalase. All right. And so that tells you a little bit about what category to put the bacteria in. Okay. Adding hydrogen peroxide. Okay. Bubbles. Okay. So your sample. Okay. There's catalase in it. All right. Here's some examples of different organisms, right? Aerobes can use oxygen in their metabolism and have enzymes to take the oxygen and convert it into something It's not harmful when it's done. Okay. An organism that can't grow without oxygen is called an obligate aerobe. Obligate means you must have oxygen to live. We are obligate aerobes. We have to have oxygen to survive. Okay. And the reason for that is you make 20 times more energy with oxygen. Okay. So that would be obligate aerobe. Okay. Obligate aerobe. There are bacteria that are obligate aerobes. Okay. You can use a medium called thioglycolate that has different oxygen concentrations at different levels of the tube, and you can see that the growth of an aerobe is going to be near the top because that's where the oxygen is. Okay. A microaerophile doesn't grow at normal concentrations of oxygen in the atmosphere, but needs a little bit of oxygen. Okay. And you can see how they would grow here. Okay. So this would be less oxygen. Right. So oxygen's high at the top and gets lower as you go down. A facultative anaerobe can switch it. Okay. It can go with or without oxygen. Okay. So it can grow throughout the tube. Okay. And we'll say variable oxygen. Okay. Variable oxygen. Okay. An anaerobe doesn't want to be with the oxygen. Okay. And so it's going to grow at the bottom of the tube where you've got very low oxygen. Okay. And an obligate anaerobe can't have any oxygen. Okay. An obligate anaerobe would grow almost at the very bottom of the tube. An aerotolerant anaerobe doesn't use oxygen, but can grow to a limited extent with it around. And they aren't harmed by oxygen because they don't have mechanisms that turn it into something that's toxic to it. Okay. And so again, you've got growth throughout the tube. Okay. So that's variable oxygen. Okay. So different amounts. All right. Let's take 10 minutes and come back at 2:05. Take a bathroom break and we'll finish up. Okay. All right, we are back. And the topic is carbon dioxide. Okay. CO2 to us is a waste gas. Okay. But to a capnophile, all right, they like CO2. They like CO2 in the atmosphere because they use it to make the things that they ultimately use to generate energy, right? So these would be things that photosynthesize, okay, or just in general, what we would call autotrophs, okay? So photosynthesis or chemosynthesis, where you pull the energy from something like a metal, okay? So important in the initial isolation of these organisms from clinical specimens that like high CO2 will include Neisseria, okay, Brucella, Strep pneumonia, okay? They all like high CO2, right? So when that's the case, when you have to grow it in high CO2, you can use an incubator. With a CO2 tank, okay? Or if you don't have something like that, you can use a little interesting trick called a candle jar, okay? So here you get a jar, okay? Just a glass jar with a lid. Okay? And you put your plates down in the bottom. They're going to grow your capnophile. Okay? They just stack. Those are plates. And then you put a candle on top and you light the candle with the lid on. So you light the candle. Okay. You got the lid on the jar. Right. And what does the candle do? The candle burns up all the oxygen. And when the flame goes out, what you have is high CO2, okay, and no oxygen, right? So that's another way around it. pH is the acidity or the basicity of a solution, and it's on a scale from 0 to 14. And basically, when you're talking about pH, you're talking about proton concentration. Okay. So at a high pH, the proton concentration is low. And at a low pH, the proton concentration is high. Okay. 7 is pure water, that's neutral. And so those are the values, right? Above 7, basic. Below 7, acidic. Okay. The organisms that we're going to be interested in are going to grow between pH 6 and 8. Why? Because that's body pH. Okay. Acidophiles like acidic environments. Some euglenas can grow between 0 and 1. Thermoplasma lives in pHs of 1 or 2. Picrophilus can grow at 7, but can live to a pH of 0. And a lot of molds and yeasts tolerate acid and are spoilage agents of pickled food. Alkalinophiles like alkaline conditions. Natromonas lives in hot pools and soils around pH 12, and Proteus can create alkaline conditions to neutralize urine and colonize and infect the urinary system. Okay, so different tools, right? So another one we should mention, that's a, we would call it a facultative acidophile, is Helicobacter. Okay, Helicobacter can survive in the pH of the stomach, which is between 1 and 2, right? But it can grow at other pHs as well. And Helicobacter causes gastric ulcers in some people. Osmotic pressure, right? Osmophiles like high solute concentrations. So let's call it salty environments. Okay. And halophiles like very high concentrations of salt, like halobacteria and halococcus, right? 25% sodium chloride is optimal, and they have to have at least 9% to survive, right? Facultative halophiles can switch off, okay? They're salt resistant, but they don't normally live in those environments. Staph is an example, okay? It can grow on media that contains sodium chloride between 0.1 all the way up to 20%, okay? Now, where do you normally find Staphylococcus? And the answer is on your skin, right? Which is high salt because of sweat, okay? And generally low moisture, right? So these guys love that, right? Radiation, okay? Radiation is a form of energy, okay? Phototrophs use visible light as an energy source, while non-photosynthetic microbes tend to be damaged by the toxic oxygen that's generated when UV hits the oxygen and turns it into those toxins, those free radical things like superoxide and so on that we talked about earlier. Some species make a yellow pigment that absorbs and dismantles toxic oxygen. Ultraviolet and ionizing radiation, as a result, can be used to control a lot of microbial growth. So this is what you would use to, say, sterilize stuff that you couldn't put in an autoclave, which has a very high heat and pressure, right? So this is how we get things like sterile plastic products, right? Hit them with the UV and nothing's going to grow, okay? Barophiles exist under pressures that range from a few times to 1,000 times the pressure of the atmosphere. They're strictly adapted to high pressure that sometimes they'll rupture if they get to atmospheric pressure, right? So this is one of the problems with organisms of the deep sea, right? When you want to study organisms that like to live in the deep sea, it's very tough to study them at the surface because of the high pressures that a lot of them live under. And what happens when you bring them up to the surface is they pop, right? And they're dead. So they need those high pressures. Other organisms are going to live in habitats with each other, okay? So we're going to have associations between either similar microbes or dissimilar microbes, right? Organisms that look and act like me and those that don't. So these can happen between animals and plants, between microbes and animals or plants. I mean, there's all kinds of different interactions that are out there, right? Some of them are beneficial, right? Some are harmful, and some have no effect, okay? And the interactions can either be obligatory, meaning they have to happen, must happen, or non-obligatory, meaning it doesn't matter whether it happens or not. And often it involves food. Okay, an example here, these birds and this, I think it's an elk, maybe. But what the birds are doing is removing parasites. From the skin, right? That's what they're doing to him or her. So the birds get food and he gets cleaned of the parasites that can spread disease. So that's a good deal, okay? In a symbiosis, both organisms benefit from the relationship, right? And that's like we saw on the previous slide. The three types are mutualism, where the organisms live in an obligatory and mutually beneficial relationship. So what would we call that? You scratch my back, and I'll scratch yours. Okay. Commensalism, where the partner that is the commensal receives the benefits while the other partner isn't harmed or benefited. And parasitism, where one organism gets all the benefits, okay, and harms the host, okay? So example of parasitic relationship would be a dog and a tick. Okay. And then commensal relationship, let's see if I can think of one here. I would say, you know, mutual benefit and all those. You know, the clownfish and the sea anemone. And you might say, what are those? Okay, well, a clownfish is a cute little fish. He's got orange and white stripes and he lives on the ocean floor. And the sea anemone is an animal that generates a toxin that the clownfish can get on its skin as it swims around the sea anemone. Okay, so the sea anemone isn't affected really by the clownfish, but the clownfish gets protection by living inside the sea anemone. Okay. Associations are a little different. Association is between free living species that come about when they compete for stuff. In antibiosis, you generate some chemical compound that is released into the environment that destroys another microbe that wants to be in the same place. Okay. The first microbe is going to have the advantage because he got there first and got a foothold. Okay. You see this in soil where mixed communities are going to be competing for space and for food. In synergism, you have an interrelationship between two organisms that benefits them, but they don't have to have it to survive. The participants cooperate to produce a result that they couldn't generate on their own. An example is dental plaque, where there are bacteria that live in your mouth that will form little communities on your teeth. Some of these bacteria are good at building a home for the community. Some are good at defending against attack, and some are good at doing things like getting nutrients. Okay, and so together, they're actually stronger than they are if they try and go it alone. And so it's thought that these were the first examples of what would eventually become tissues early in evolution. Okay, so synergism. So that's a biofilm, right? You got mixed communities of bacteria. A colonizer comes in, gets on the surface first makes a secretion. Other microbes hook onto that and secrete other material, and the attached cells are stimulated to release more chemicals as the population grows. And then they even make structures out of protein that allow them to detect changes in their environment that they can communicate to all of the members of the biofilm. Okay. The members of the biofilm have different characteristics. All right. The bottom of the biofilm may have different oxygen conditions and pHs than those at the surface, and they form partnerships that allow them to prosper that wouldn't happen if they were alone. Okay. So they're tough to wipe out using traditional methods because it's not one kind of pathogen, right? It could be several. Bacteria and biofilms behave and respond differently than free living bacteria because of different genes being used in the biofilm, and they behave differently. Okay, so they're tough to knock out. So that's why the dentist is doing all that scraping when you go in for your cleaning, is he's getting the plaque off of your teeth. It's difficult to like gargle with an antibiotic that would knock out all the things that would participate in the plaque. Okay. So instead we go to mechanical processes like brushing and flossing and scraping to get the stuff off. Okay. And then we'll talk about here a growth curve. Okay. So bacteria are interesting because under the right conditions, they grow real quick. Okay. If you give the perfect conditions, for instance, to E. coli, which lives in your colon, it can double every 20 minutes. That's pretty fast, right? Compared to how quickly we reproduce. The process is called binary fission. Okay. So this is bacterial mitosis. Okay. But it's a different process than mitosis because it doesn't involve chromatin. And you're only talking about, in most cases, one chromosome. Okay. So what happens is that the parent cell gets bigger, it duplicates its chromosome, and then starts to pull its envelope together in the center of the cell, and then forms a new cell wall, and then we split. Okay. In most cases, this is very rapid. Okay. Rapid reproduction. The generation or the doubling time is the amount of time it takes to go one round of reproduction. Okay. So we increase the population by a factor of 2. If the environment is always good, the doubling time can continue at a constant rate. Okay. And there are ways to set that up. The length of the generation time is a measure of the growth rate of an organism, how fast it reproduces. Okay. Average generation time is between half an hour to an hour. Shortest generation times between 10 and 12 minutes. Okay. Mycobacterium, which is associated with leprosy, has a generation time of between 2 weeks to a month. So it's a slow grower. Environmental bacteria have generation times that are measured in months as well. The pathogens have a short generation time, and that's how they produce disease. Okay? So you can see some examples of how this works, right? You can start with one cell, and then after one generation, we've got 2 generations, 4, after 3 generations, 8, after 4 generations, 16, and after 5 generations, 32. Okay. So up and up we go. All right. And so we can plot those changes in numbers. If we were to take these numbers and draw them on a regular graph paper, it would look like this, right? We start with these low numbers and then flammo, we take off. So we make this curve, and this is what's called exponential growth. Okay. So this is very fast. Okay. Very fast. That's why the line almost goes straight up, right? Line's going straight up. That's a lot of speed, okay? Or a very high rate. So sometimes if we want to show this in the form of a line, we can use what's called semi-log paper, where you've got one axis that's like a normal axis, where the increments are always the same, right? 5 to 0 to 5, 5 from 5 to 10, and so on. And then on the vertical axis, you have a logarithmic scale, okay, where each of these varies by a factor of 10, right? So the divisions here may be units, the divisions here may be tens, the divisions here may be hundreds, and the divisions here may be thousands, right? And that way you can draw a straight line on this graph that you can write an equation for. Remember, straight lines are in the form of y equals mx plus b, right? This is going back to algebra where m is the slope, okay? And b is what's called the y-intercept. Okay. And then y is just the point on the line that's the y coordinate, and x is the point on the line that's the x coordinate. Okay, that's the formula for a straight line. So if you have a formula like that, you can plug in a value from the y-axis and get a value from the x, or you could do the reverse. Okay, so if we use this equation, all right, the size of a population can be determined at any particular time, all right? Where this figure here, n sub t, is the number of cells in the population at the time t, okay? So that could be days, that could be minutes, that could be hours, okay? And then n sub I is the inoculum. That's how many cells you started with, okay? And that would be a known number. And then 2 to the n is the number of generations. Okay, number of generations. Okay. And 2 to the n represents the number of cells in that generation. Okay. A growth curve is just a way to show how the growth would happen in a particular set of conditions. And so we place a number of cells into a liquid medium. We incubate the culture for some amount of time. We take samples at regular intervals, and we plate the sample onto media, and we count the number of colonies you've got, and then we can make a curve from that. Okay, so here we go. Like we start, all right, we put some number of cells in here, okay? Like one colony forming unit, like could be a pick off of a plate or something, okay, of a colony. And that's going to be about 5,000 cells, right? Okay. And then an hour later, we stop the shake, we take a sample, we plate that. An hour after that, we stop the shake and take a little bit and plate that, and so on, and so on, and so on, right? And the number of cells growing on the plate each time is going to increase, right? And we can work backwards from the number of cells on the plate to figure out the number of cells that are actually in the tube, right? And then we can make a plot from there, right? So that's our estimate that's in the tube, right? And that's the number of colonies on the plate. Okay, so you just count them and you work backwards and you use that same formula, okay? So if we look at a typical growth curve, newly inoculated cells need a little bit of time to get going, all right? Cells aren't yet reproducing at their maximum rate, and the population of cells is tiny, so sampling may miss them completely. In the exponential phase, the cells are growing very rapidly, okay? And that will continue as long as the cells have something to eat, all right? In the stationary phase, the rate of reproduction equals the death rate, okay? So you won't see an increase in the number of cells here. Cell division slowing down, junk is building up inside the flask that actually keeps the growth rate getting lower, all right? And then in the death phase, the cells start to die at an exponential rate because of the waste products that build up. That speeds up even more as time goes by. It's usually slower than the growth phase, okay? And eventually we have everybody dead, all right? A viable non-cultural state is where a lot of cells in a culture are in the death phase. They stay alive, but they're dormant, okay? So they may be spores, all right? Or they may just be sleeping, okay? But they're still alive. So you can see what that looks like here, right? Exponential growth, stationary, death, right? But some cells are still alive, but they don't grow on a plate, right? So here, birth rate is greater than death rate, right? Here, birth rate equals death rate, okay? And here, birth rate is less than death rate. Okay? And finally get to the end, right? So the whole time, you're depleting nutrients and generating waste. Okay. So that's what a growth curve would look like, right? This is a log axis here, right? And this is a regular axis. This is time. So the tendency for a population to show different parts of the growth curve is important, right? Microbes in exponential growth are going to be easier to kill with antimicrobials or with heat or with antibiotics. Why is that? Well, more growth means more synthesis. Which means more damage potential. Okay. So it's when you're putting stuff together, it's easiest to wreck it. Okay. Actively growing cells are more vulnerable to conditions that disrupt cell metabolism and binary fission. a person actively shedding bacteria in early and middle stages of an infection is more likely to spread than a person in the later stages, okay? Sometimes we want to keep these perfect growth conditions always, all right? And so we can use something called a chemostat that allows old nutrients to keep coming in and removes used media and dead cells so they don't ruin what's going on in the chamber, all right? So the growth rate stays at that perfect level in the exponential phase, right? And we can use what grows in there for industrial applications, like to make particular drugs or to break down certain things and remove the byproducts, okay? So the idea there is a constant perfect environment. There's another way to determine your growth curve, right? One way is called turbidity, or we could say cloudiness. Cloudiness. There we go. The cloudier it is, the more stuff is in your media, okay? To the point where you won't be able to see through it anymore, right? So the population increases as the turbidity goes up. And then there's counting, where we actually take a little bit of the media and we put it under a microscope and we stain it, and then we count the cells, and then we calculate how many were actually in the container from that count. A Coulter counter is going to do this electronically, right? While direct count uses microscope in your eyeballs and a clicker. A flow cytometer is like a Coulter counter, but it can measure cell size and it can tell live cells from dead ones. So that's kind of nice because that gives you really a more accurate idea of what's alive in the culture at any particular time. Genetic probing uses PCR to determine how much growth is going on. PCR is just a way to determine how much DNA is in a sample, okay, in this case. And the more DNA is in a sample, that means the more cells are in the sample, and you can turn around and say that that is a reflection of your growth rate, okay? So this is direct counting. Okay, and this is flow cytometry. Okay, and it's all to get an idea of how your organisms grow in culture in a lab, and that makes it easier for them to study, and it makes it easier for us to figure out, let's say, if they're a pathogen, how to kill them, right? Or if they're doing something good for us, how to make them grow at the best possible conditions for the longest time so that we can pull off what they're making, right? So let's discard what we wrote down. Next thing we want to talk about is lab today, okay, which is going to involve something called Kirby-Bauer. So let's pull this up. Yeah, probably. All right, let's go to here. Hush. Columns. There we go. Here. There we go. Yeah, that's why that's in there that way. We want. Let's just go. There we go. There we are. Okay. Yes. I'll find it in a second. Sensitivity. There it is. Okay, so. Okay, so here. I'm gonna go here. There we go. I'll open with this guy. Share. Okay, here we go.
What we're going to do on this test is antibiotic sensitivity. In order to do that, we need a TSA plate and we need a bunch of antibiotics on little filter paper discs. And they're actually impregnated with a given amount of an antibiotic, and the discs are marked. In this case, even though you can't read it, this one says VA30, which stands for 30 micrograms of vancomycin. We're going to put these onto the plates. The tube normally shows up in a dispenser like this. Right? The dispenser holds 6, 3 on this side and 3 on this side of these dispensers. When pushed, this will dispense a set of filter paper discs onto our plate. You're going to take a plate, take an organism, you're going to flame your loop, cool your loop, and spread the organism over the entire surface of the plate. Just keep going until you've covered the plate completely. You will notice that I've turned this multiple times to keep going back and forth. Oh yeah, don't talk on the plate while you're doing it. And then flame your loop, cool your loop. That part's done, right? Once you're done with that, put it lid down and write on the plate what the organism is and your initials. Do it now, because once you get the filter paper discs on, you got to leave it right side up. If you don't leave it right side up, the discs fall off. If the discs fall off, it doesn't work so well. Once you're done with that, you need to apply the discs. And how are we going to do that? Turn it right side up. Take the lid off. We set our dispenser right over the top. Remember, I took the lid off. Otherwise, the disc just wind up on the lid and it does us no good at all. And I just simply push down, release, move the dispenser, put the lid back on, see our discs sitting on here, and we will simply incubate this right side up. In this one here, you will notice that we don't have anything growing except for one big fuzzy, one big mole growing on this. Well, one big mole growing on this isn't going to help us much. This plate was not inoculated at all. It was just used as a demo like the other one. The thing is, all of these are antibacterial antibiotics. And obviously, considering the fuzzy's growing right on top of the disc, they don't work so hot on fungus. So in order to do something against fungi, we would have to do this with a bunch of antifungal discs instead of antibacterial discs. Here's what one winds up looking like if we can zoom in on it just a little bit. This one's been incubated. And what you'll notice is, in this case, this one that says AM10 or ampicillin 10 micrograms, it doesn't affect it at all, does it? The organism is resistant. And as we go around, you can see slightly bigger areas, clear areas around the outside. Clear areas are zones of inhibition, as they're called. And one would think that the bigger it is, the better it is. Well, not necessarily. Some of the antibiotics are smaller in size than others. The smaller they are, the better they move out from the disc out through the media or out through the agar. So sometimes the bigger zone doesn't mean that it works better, it just means that it's smaller. Maybe this one happens to be a very big antibiotic and it actually works better than this one here, which might be a very small antibiotic. But in order to determine whether the organism is resistant or sensitive, you just simply measure the distance from the disc across clear area and then compare it with a table in your book that gives you what the standards are supposed to be. And you can determine antibiotic sensitivity or resistance to various antibiotics. Same way they do it in the lab. So you may not be doing this in a lab, but you're going to be getting a report sheet out on this. And it might be nice to know what's going on.
Okay, so if we look here, the report that I'm going to send you that deals with Kirby Bauer, the part that you want to do is called antimicrobial sensitivity, right? So you hit this, okay, and then you're going to hit the Kirby Bauer link, okay, and then you're going to have to open it. I open it with Flash Player. So there's a link to downloading Flash Player right here, okay. I know that Flash Player on mine is located here. Okay. I'll open that. Okay. here. There we go. Now we've begun. So now let me show you. New share. And here, right? So that's what it looks like opened in Flash Player. And you just hit start, okay? And it's just going to show you a dish with the zones of inhibition, right? And it's going to actually measure those for you, right? It'll give you numbers, okay? There's your standards for resistant, intermediate, and susceptible, right? And then you just measure with your ruler by grabbing it, move it around, what you actually got on the plate, right? And then you compare those numbers to these numbers, and then you determine whether you're sensitive or resistant or intermediate by just clicking the box, okay? You just click the box, and when you hit the right box, it'll go green for you, okay? And then you can take that data and you can put it on the report that I gave you, all right? And then that'll help you determine each of these different sensitivities, right? You can go back to the menu and you can do a restart, right? And it'll show you a different plate, a different organism, right? This is Pseudomonas aeruginosa, right? It says so right up there. And then you can go through and do the same thing with these, all right? Figure out who grew and who didn't, right? Hit menu again, restart. There's Staphylococcus aureus, okay? Do the same thing. Go back to menu, restart. There's E. coli, that's the first one we had. So there's 3 plates, 3 sets of data. And just enter those on the form and then answer the questions that follow, right? Who was sensitive, who was resistant, and to what, right? And then there might be a question on there as to why something was sensitive or resistant to a particular drug. And for that, what you want to do is look up what the antibiotic is. So you can Google that, go to like Wikipedia, and it'll talk a little bit about the drug and how it works, okay? And if one of the drugs didn't work, you can figure out why, right? And that'll mean something for that particular bacteria, right? So if I was just, like, let's say I want to treat an E. coli infection and I had these data, I would probably pick chloramphenicol just based on size. But I actually have to go to this data to determine whether I made the right choice, right? I'd have to measure this, right? Compare it to the data in the table and see if that's the best choice for the drug. And the idea behind using this is that you always want to use the minimum amount of drug that gives you the maximum effect. And the reason for that is that you don't want to just be giving your patient tons and tons of an antibiotic that their liver has to process, because you'll hurt their liver. You want to give them the amount that's going to wipe out the infection, but be easy on the rest of their organs. Okay, so that's why we do a test like this, right? And the website is in the email that I sent you from last week. It's the same website, and you'll have the report, and it's due next time we meet by that Wednesday. Okay, and I will work on getting you those codes so that your Connect doesn't get shut off. All right. And what they'll probably do is just move you from the 2-week temporary right into permanent so that you can finish your homework. And then you want to complete this. And I'm also going to put up practice exams for you to get you ready for the weekly exams and for the midterm and the final. Okay. And those won't have any point value, but they will help you prepare, right, to get ready for the questions that are on the real deal. And that
Mr. Saunders, how are we using that ruler again?
Okay, so you click and you grab the ruler, and you're just measuring from the edge of the disc to the edge of the clear zone.
Okay.
Okay, and then you just compare that number to the numbers in the table, right? So these are the numbers that are sensitivity. This is intermediate, and this is resistant, okay? And these are all in centimeters, or sorry, millimeters, okay? And then see the little dots, the little divisions are in millimeters. So if I was to measure this one, okay, this would be 1 point, looks like 2 centimeters, okay, which would be 120 millimeters, right? So I can go here and, I'm sorry, it looks like 12 millimeters, right? So for C, which is chloramphenicol, I'm at 12. So this would be a poor choice actually, because this is resistant, okay? So that's how you do it. You just measure, record the number, and then figure out who's resistant. But see if you click, right? Look at that, click there, that's a no-go. That's a no-go. Okay. And then that there is a susceptible, right? So they're saying that's susceptible, that's greater than 18. Let's see, maybe it's the center of the disc. Must be. Okay. But it'll tell you as you click through it what the numbers are. Okay. So it's pretty straightforward, right? If you did this in a real lab, what we would do is we would have you guys grow a lawn of these different bacteria, get that lawn up and healthy. And then the following week, we would put the discs on with the little disc dispenser. And then we would let some time go by, and then we would read them, okay, and compare them to what's in the table. And that'll let you know. So when the thing glows green, that means you've got the right choice, okay,
Gotcha.
on the animation. Okay, take that down, right? And other than that, do we have any questions? You good? Okay. Yes.
Are you gonna send the access? Because I didn't receive it yet.
Yeah,
The access code.
right. I have to get that from from our representative. Okay.
Okay.
Yeah.
All right, thank you.
All right, guys. I will see you all either in tutoring on Friday, or I will see you next week. Thank y'all for coming. Appreciate it.
Okay, thank you.
Mhm.
Thank you.